Go ahead and get started. This is our last Monday, July 26th of what has been both an incredibly long and an incredibly fast summer session, as I'm sure you feel that as well as I do. We have one last major topic we need to talk about, and that is going to be the autonomic nervous system. So that's going to be our focus for today and tomorrow. Both the anatomy, the physiology, all of that kind of stuff. And like I said, I know it's the end of the semester. I know we're exhausted. I know you're tired, but as I mentioned, this stuff we're going to cover over these next 2 days is probably the most important material you're going to need moving forward into 431. So obviously you need to know your tissues. Obviously you need to know your regional terms. You need to know all of that stuff as well. But this stuff is how we control all those organ systems we're going to be talking about in 431. So it is important. You do still have 2 more assignments due tomorrow. Unit 14 review, which you know how to do. That's simple. The reflex lab, remember you're just doing procedures 2 on pages 371 through 374 and procedure 2 on page 380. And again, try to do them to your best of the ability. I know you're not going to have all of the resources necessary to complete those. So anything that you aren't able to complete, then just do it as a thought experiment. I want answers of what you think would happen or how it would occur. Again, I don't want any, I didn't have, X, Y, and Z, so I didn't do this activity. That's not what I want. I want you to at least pretend that you could do it and think what you would, based on the information you have, what you think would happen. All right. Thursday, the 29th, apparently we have a lab and lecture exam. So that should keep you pretty busy on that day. Again, remember that is just going to be like the previous 3 you've taken on and will just involve the nervous system, just like we've seen with the other sections. So again, you can take them in any order and everything's the same on those as the previous ones you've taken. And then Friday, you will have that 100-point cumulative final exam, which again is all multiple choice questions. You don't have to pull the information out of the ether. You just have to recognize it on the list of possible correct answers in front of you. And it covers everything we've talked about in this class. All right. So, yeah. Yes.
Allotment for the 100-point final.
I'm sorry, say that again.
What's the time allotment for Friday's exam?
2 hours. So you have basically 100 minutes to answer 100 questions. So it's going to be 2 hours. So with the setup, with the scanning of your room, all of that kind of stuff. So again, it's just like the final exam that you would take in the classroom. You have a 2-hour window of time. And normally I'm supposed to give you just a 2-hour minute, so just say between 8 and 10 is when you have to complete this. I don't think that is necessary. So again, you can start it anytime during our normal class time, but you will have 120 minutes essentially to be able to complete all of that activity, which should be more than enough time.
Okay, so it will still be available between 8 and 12:30. We just have 2 hours to complete.
Yes, I know I'm supposed to narrow the time for that, but I don't think that that's necessary in this case. So you will be able, but again, make sure you don't wait till noon to start it, because remember, it still must be completed during the class time. So if you wait till noon to start it, you'll only have 35 minutes to be able to complete it. But yes, if you want to wait till 10 to start it so you can sleep in or study a little bit more in the morning or something like that, then that should give you more than enough time. So yes.
Perfect. Thank you.
All right. No, great question. Any others?
Professor?
Yes.
So for the 100-point final exam, since it's a multiple choice, would we know our scores right after we submit them, or would they take a while?
Okay, great question. So no, you will not be able to view your score immediately after completing the final exam. Again, anytime you have a test bank with this many questions on it, there are bound to be errors and stuff like that. So I like to review it before I post the scores. And even if you know the score of the final exam from Friday, I still obviously, by Friday, I won't have your lab and lecture exams from Thursday graded yet. So I won't have your final exam. So that's actually a great question. My goal is to, by maybe Wednesday, what day of the week would that be? So I'm hoping my goal will be by Wednesday the 4th. All the grading will be done, and final grades will be posted. That should be my goal, and I'm hoping that we'll be able to do that. If that's the case, I'll get them posted. And again, just to be clear, I will be posting these on Canvas, is what I mean by that. Then you will have 2 days to review your scores, ask any questions, check to make sure everything's up to date. And again, you should have been following these things along because all your grades have been available up to this period of time. But if there's any questions or anything like that, you will have a couple days to be able to address those with me. And then on Friday, that would be probably the 6th, I will finalize the grades and submit them to e-services. So that will be the game plan. So I'm hoping to start on Thursday, start grading those through Friday and the weekend while you guys are working on that. So as soon as I get them graded, Wednesday would probably be the latest. I'm hoping to get it done before then, but you'll have a couple days. I'll post them. I'll send the announcement that they've been posted, and then you'll have a couple days to review everything before I finalize the grade in e-services. Okay, so that's my schedule, although I'm hoping to get it done before then.
Okay,
And
thank you so much.
No, thank you. Yes.
Sorry, the Wednesday study day extra credit, that'll be, does he send over the information or hours?
Absolutely. So Jeff, well, I assume this week, I assume he's still available this week as well. So this weekend, I will email Jeff and he will email me the list, and I will, in fact, I'll send him an email to remind him that I need that. So yes, I will email, Jeff will email that to me, or if he doesn't, I will remind him to email that to me. That purposely has not been added in. In fact, if you look at your grades, you don't actually see a spot for that in your grades, because again, I don't want you counting on that. I want you getting the grade that you want in this class without it. And if you fall that tiny bit short, then hopefully things like the open lab can be enough to put you over the top. All righty. Excellent questions. I like it. Any others?
I don't know if anybody asked. I was looking up the procedure thing, but will the final be curved, or is that just straight? What you get is what you get.
Unlike the other exams, typically I don't curve it as a default. All the other ones by default I curve. The final, I figure it's 100 points, so it should be, 100 questions should be 100 points. If for some reason, like I said, I'm constantly adding and adjusting questions to the test bank for that. So if it turns out that something unusual happens or there's an odd number of confusing questions, or for some reason a class does particularly poorly, I reserve the right to curve it if needed, but I would say in most cases I haven't needed to curve it. I would say the averages on it are at least as good as for the normal lecture exams. So it often doesn't require a curve, but if something unusual happens, then I'm not opposed to curving it if needed. Right. All righty. Any other questions?
The reflex lab, you just want that just on a regular old word processor or Word doc or something like that?
You can do it that way, but again, remember, it's out of your lab manuals. So the same way you do your reviews out of the pages of the lab manual, you can just fill in the pages of your review sheets. So it's right out of the lab manual, so you can do it the same way. I don't think there's any drawing for that one. So I guess there's no reason you couldn't do it on a separate page, but if you have the sheet, then why recreate the wheel? But yeah, so that should be fine.
Okay.
All righty. Out of my way. Any other questions? Awesome. All right, let us dive into lecture then. As I mentioned, our last big topic that we have to talk about for our 430 class is the autonomic nervous system. We've already talked about our autonomic nervous system already. Autonomic nervous system is primarily controlled and involved in controlling the activity of what type of effectors?
Sympathetic and parasympathetic.
Well, those are the branches, but what does it actually control? What are the
Smooth
targets?
muscle, cardiac, and glands.
Yeah, there you go. Exactly. Smooth muscle, cardiac muscle, and glands, right? It's what provides us with that involuntary control, right? Excellent, excellent, excellent. And of course, as we've talked about then, that involuntary control, why do we control our stomach? Why do we control our heart? Why do we control our sweat glands?
Homeostasis.
Yeah, it's about maintaining homeostasis, right? And it's involuntary. It isn't something that we have to consciously think about. So it basically uses that concept we talked about in the last class, like that reflex arc, to basically do this job. There's some type of sensory structure that perceives some type of change going on in the body. It sends that information to our command center where a decision is made on that information. And again, we're not talking about the cerebrum in this case because we're not voluntarily making a decision on this, but a decision is made, synapses are made, postsynaptic potentials are counted, and then a signal is sent out to our smooth muscle, our cardiac muscle, and our glands to tell them what to do. Of course, that means we need sensory information. And one of the important things to remember about our autonomic nervous system is that it doesn't just rely on autonomic sensory information. It receives information from all types of sensory information, right? Yes, if the level of calcium in my blood drops, then I need to make some changes in my body to bring that up. Or if my internal body temperature drops, I need to do something to bring it back up. So definitely those are things that are going on. However, can things like changes in body temperature affect your heart rate or your respiration rate?
Yes.
Yeah, when your body increases in temperature, what happens to your heart rate?
It goes up.
Yeah, it goes up. It's one of the problems with those polar plunges, right? In places like Chicago, right, on the winter solstice, one of the things that they all do is go out and jump into the river. Right, it's a big, huge thing they do in Chicago, and there's several other locations where they do these polar plunges. And that's a problem. You jump into this freezing cold water and your temperature drops dramatically. And there is the potential that that can actually cause your heart rate to drop dramatically and actually stop your heart from beating. Right, so that temperature sensation or pain or other types of somatic reception have the ability. And when you're sitting enjoying your backyard and your next door neighbor has that tri-tip that they've been marinating for the past 3 days and it hits the grill and you hear that sizzle and you smell that, assuming you're not vegetarian, smell that the smells of that tri-tip coming across, does that olfactory sense or that visual sense or the hearing of the sizzle have the ability to say, I don't know, affect your salivary gland production?
Yes.
Yeah, absolutely. So notice we take in all sorts of different types of information. So when we talk about our autonomic nervous system, what we're really talking about is a motor output, because we're taking information in from all parts of our body to process. Of course, we need a place to process it, and it is primarily processed in the nuclei, those clusters of cell bodies in the medulla and in the hypothalamus. Remember, this is not up in the cerebrum because this is not something we are voluntarily controlling. But typically when we talk about the autonomic nervous system, we are talking about a motor output system. All sensory information comes in, it is processed in our central nervous system. And so when we're talking about our autonomic nervous system, we are talking about a motor output system. And for us, this is a very good thing because we've already learned about a motor output system, right? We have learned about the somatic motor nervous system. And that's something we're familiar with. Let's go to our whiteboard. And draw a big fat line right down the center. We have learned our somatic motor pathway. Now let's start with some of the easy things. What, of course, is the effector of our somatic motor pathway?
Ventral root.
Well, again, what is the effector? In our somatic motor pathway, what is, there we go, Samuel's got it, skeletal muscle. Our goal is to get to that skeletal muscle. So let's draw a little cylinder-shaped cell here. Heck, we'll even give it some stripes. Our goal is to get to that skeletal muscle. Now, we know this has to start in our central nervous system. And I'll give you a hint for our somatic motor pathway that begins in the spinal cord. But let's be more specific. Where in our spinal cord does our somatic motor pathway begin? Where is our cell body located?
Anterior gray horn.
Excellent. Yep. Brian and Samuel got that for us. And of course, as you know, the cell body that is located in this anterior gray horn, this somatic motor neuron, what is its structural classification?
Multipolar.
Multipolar, excellent. We know its axon's going to come out the ventral root and basically travel all the way to our skeletal muscle cell, where it's going to cheat and just do that, form a synapse. What do you think? Myelinated or unmyelinated axon?
Myelinated.
Yep, myelinated indeed. We hadn't talked about that yet, but I figure we know most axons are myelinated, so I'll cheat and put some myelin on this one. And we know two other things. What neurotransmitter is released? Excellent. Acetylcholine. And, oops, I need for that C to be capitalized. And what is the effect of that neurotransmitter?
Excitatory.
There you go. Perfect. There you go. Notice absolutely positively no new information here on the screen, just a summary of things we've already known. Heck, way back in the muscular system, we learned about how that axon releases at its synaptic end bulb acetylcholine. It has an excitatory effect. Right now we know that that cell body is actually located in the anterior gray horn of the spinal cord. So we have all of this information and we're comfortable with it. And again, it's awesome because there's nothing new there. And so it's just a good reminder. And now we can use what we know to compare it to our autonomic motor pathway. Again, remember when we say autonomic motor, it's autonomic pathway, it's always motor. But putting the motor there is a good reminder that it's an outside pathway going out of the central nervous system. And once again, let's start easy with what we know the effectors are going to be. And I know you guys already told me this just a minute ago, but let's go over it again. What are the effectors?
Smooth and cardiac muscle and glands.
There you go. Cardiac muscle, smooth muscle, and glands. Excellent. So that is going to be our effectors. And heck, we drew the skeletal muscle. So here's my branching cardiac muscle. Here is my spindle shape, smooth muscle, and we'll put a couple blobs here to indicate a gland. Excellent. Now, this motor pathway also starts in the central nervous system and also starts in the spinal cord. So once again, we have a central nervous system start. However, this pathway begins with a cell body located where?
The lateral gray horn.
In the lateral gray horn. What is, oh, we didn't do that for this one. What is the structural classification of this neuron?
Multiple.
Polar.
Excellent. It is a multipolar neuron. Its axon, does it go out the dorsal root or the ventral root?
Ventral.
Yeah, ventral root, excellent. And what do you think? Do you think it's myelinated or unmyelinated?
Myelinated.
Yep, it is indeed myelinated. So other than the cell body location, we're pretty similar in the way this is set up so far, right? And that's fun. But here's where something interesting happens. Unlike up here in the somatic motor nervous system, where it is just a one neuron pathway from central nervous system to effector, in our autonomic nervous system, it is a two neuron pathway from central nervous system to effector. So this neuron here that started in the central nervous system doesn't make it all the way to the effectors. Instead, it synapses on a second autonomic motor neuron. And of course, still an autonomic motor neuron, so it is going to be multipolar. And it will be this second neuron that gets to go and innervate the effectors. All right. Now, two things. We know that cell bodies can't just be hanging out all by themselves. So they have to cluster together in a structure. And what do we call these structures that are a collection of cell bodies outside the central nervous system and the peripheral nervous system? There you go, same as got it. This is going to be an autonomic ganglion. So this autonomic ganglion is where the second cell is going to be located. Now, anytime we have two neurons in a pathway, we need to make sure that we are able to distinguish the two of them. A and B, 1 and 2, that would be a really nice, simple way to do that, but we know anatomists hate us. So that can't be that simple. Instead, they identify them by their relationship to this ganglion. So that means the one that starts in the central nervous system and goes to, or let's say it this way, synapses in the ganglion. We call this one the pre-ganglionic neuron. This is a little bigger than it needs to be. So our preganglionic neuron is the one whose cell body starts in the central nervous system. Axon comes out the ventral root. Its axon is myelinated. It's a multipolar neuron. Its cell body is located in the lateral gray horn. And it turns out we know a couple other things about it as well. Excuse me.
Pleasure.
All preganglionic neurons release our neurotransmitter acetylcholine, and they are always excitatory. All righty. So far, so good?
Yep.
Excellent. And being the smart, sophisticated students you are, if the first neuron is called a preganglionic neuron, what do you think we call this second neuron?
Postganglionic.
There we go. So our postganglionic neuron is the one that starts in the autonomic ganglion. And synapses on the effectors. Its cell body is located in the autonomic ganglion. It is also a multipolar neuron because it is still a motor neuron. All right, this is a motor neuron carrying information to an effector, so it is multipolar. However, its axon is unmyelinated. So its axon is unmyelinated, unlike the preganglionic neuron. And there's one other big difference. Some postganglionic neurons release acetylcholine as their neurotransmitter. but some release norepinephrine as their neurotransmitter. Now, again, if we were holding one autonomic postganglionic neuron in our hand, on Mondays, does it release acetylcholine, and on Tuesdays, it releases norepinephrine?
No.
No, of course not. It's just you've got millions of these in your body, and some release acetylcholine, which notice we can abbreviate to ACH once we've written it out once. And norepinephrine can be abbreviated as NE once you've written it out once. Norepinephrine and epinephrine are two primary stress hormones. As we talked about in the last class, epinephrine and norepinephrine are also known as what?
Adrenaline.
Yeah, adrenaline and noradrenaline, absolutely. From our standpoint, while chemically there are minor differences between them, functionally they do literally the exact same thing. So basically, functionally they're interchangeable, but as it turns out, some of our neurons will release this norepinephrine as their neurotransmitter. And as we've talked about, if you sprinkle acetylcholine on skeletal muscle, it's always excitatory. But what happened when you sprinkled acetylcholine onto cardiac muscle? Did it stimulate it or did it inhibit it?
Inhibitory.
It was inhibitory, absolutely. So not surprisingly, whereas up here, the effect is always excitatory. I guess we should have thrown that in there. Down here, some of the effects are excitatory and some are inhibitory. Now, again, does that mean that on Monday the effect is going to be excitatory and on Tuesday the effect is going to be inhibitory?
No.
No, it means that for some effectors, when you release acetylcholine on them, like our cardiac muscle, it inhibits their activity. However, if a different postsynaptic ganglion, for instance, releases acetylcholine on, I don't know, our sweat gland, for instance, it's going to produce more sweat. So again, different effects on different effectors, different neurotransmitters released on different effectors. So two possible neurotransmitters that can be used and two possible effects that we can mix and match. And again, think of cardiac muscle. We already know when we release acetylcholine on it, it inhibits it. But we know norepinephrine is basically adrenaline. And when you sprinkle adrenaline on the heart, what happens to your heart rate?
Speeds it up.
It's going to speed it up, exactly. So notice by having two possible neurotransmitters that I can release to the heart, I can control my heart rate. If I need it to go a little bit faster, I can use adrenaline or norepinephrine to increase it. If I need to slow it down, I can use acetylcholine to slow it down. So it's going to give me that precise control of my heart rate by having different neurotransmitters that can have different effects. Excellent. Questions on that? All right, I've done an amazing job of drawing this, but let's talk about this again here. Again, big differences between our somatic and our autonomic motor pathways is their effectors. Somatic, it's skeletal muscle. Autonomic, it's smooth muscle, cardiac muscle, and glands. Pathways, both where they start in the central nervous system. Somatic is in the anterior gray horn. Autonomic is in the lateral gray horn. But also somatic just needs one neuron to get from central nervous system to effector, whereas our autonomic needs two to get from central nervous system to effector. And then as we talked about, they're also different in their neurotransmitters. Somatic always releases acetylcholine and it is always excitatory, which is also true for our pre-ganglionic neuron. It always releases acetylcholine and it is always excitatory. Where it's different is in the postsynaptic or postganglionic. In that postganglionic neuron, it can have one of two neurotransmitters that it can release, either acetylcholine or norepinephrine. And on some effectors, it is going to be excitatory, and on other effectors, they're going to be inhibitory. So here in words, we sum up what we just talked about, but we also have the pretty pictures as well. Here is that, again, known somatic motor pathway. We have a single neuron, multipolar, whose cell body is located in the anterior gray horn. Its myelinated axon comes out the ventral root, travels all the way to our skeletal muscle, where it releases acetylcholine and is always excitatory. Conversely, in our autonomic pathway, it's going to our smooth muscle, our cardiac muscle, and our glands. Our postganglionic neuron can release either acetylcholine or norepinephrine. But notice, as we mentioned, our preganglionic neuron always releases acetylcholine, and it is always excitatory. Whereas here, the effect can be excitatory or inhibitory, depending on the neurotransmitter and depending on the effector. Oh, I like that. Let's write that. Excellent. So what effect it's going to have is going to be determined by two things. First, what neurotransmitter is released, but also what the effector is. We already talked about the example of acetylcholine. I release acetylcholine on our heart, and it decreases the heart rate, but on a sweat gland, it makes it more active. It's the same thing for norepinephrine. I release that norepinephrine on my heart, and my heart beats faster because that's what happens when I get stressed. But how much saliva do you produce when you are stressed?
Not much. Your mouth
As you're getting ready to stand up to give that speech at the end of your English class, that's worth 45% of your grade. And as you stand up to do that and your heart is beating, your mouth is dry as a stone, right? So again, notice that norepinephrine on a salivary gland inhibits it, decreases saliva production. So notice what the effect is going to be is both the neurotransmitter, but also what the effector is as well. So both of those determine what the effect is going to be. And again, finally, in that autonomic pathway, we have two neurons, a preganglionic neuron, which again goes from central nervous system to the ganglion, and our postganglionic neuron that goes from the ganglion to the effector. And one last thing, remember, not only are they different in the neurotransmitter they can release, but remember also my preganglionic neuron is myelinated on its axon, whereas my postganglionic neuron is unmyelinated on its axon. And because again, it is important for the repetition, notice we have one more pretty picture that does a good job of showing this, plus hinting at some new material we're going to have to talk about. Notice up here is my somatic motor pathway. Single neuron from central nervous system to effector. That effector is skeletal muscle. Its axon is myelinated. It releases acetylcholine and it is excitatory. Here we have our ganglia. So we have a preganglionic neuron whose cell body is myelinated, always releases acetylcholine, always excitatory. Postganglionic neurons that start in the ganglion and go to our effectors, which are the smooth muscle, the cardiac muscle, and the glands. Some release acetylcholine, some release norepinephrine, and depending on the effector, it can be excitatory or inhibitory. Notice also, even though we haven't talked about it yet, as you also know, when you have that stress response, your nervous system can release adrenaline, epinephrine into the blood, giving us that big global stress response. Because once that adrenaline gets in your blood, where can it go?
Your heart.
True, but does it go just to your heart?
Everywhere.
It goes everywhere, absolutely, giving you a big global effect. Excellent. So again, great job of an overview of this. However, if you notice, there is two branches of our autonomic nervous system, and there are going to be some subtle and not so subtle differences between them as well. And so now that we understand the difference between our autonomic nervous system and our somatic motor nervous system, we need to talk about those two divisions of the autonomic nervous system so we can see how they're similar and how they're different. All right, so any questions on the comparison of those two before we move forward? All right, excellent. Let's talk about our two divisions of the autonomic nervous system. Of course, what are the two divisions of the autonomic nervous system?
Sympathetic, parasympathetic.
Yeah, there we go. Sympathetic and parasympathetic. And as we've also talked about, there's some cutesy little mnemonics we can use to remember them. What is the cutesy mnemonic for the sympathetic nervous system?
Fight or flight.
There we go. Which is actually a pretty good mnemonic to help us to remember that. What's the cutesy little rhyme for the parasympathetic?
Rest or digest.
We will see this one's not quite as good at really implicating what the parasympathetic does, but there are some other mnemonics we can use as well. We'll get to those in a minute. Now, one of the important things to remember is that most of the effectors, and let's take the example of our heart, have dual innervation. What do I mean when I say dual innervation?
They can have two states of innervation,
True, and what do you think those two states would be?
inhibitory and excitatory.
True, absolutely. Although another way to think of that would be sympathetic and parasympathetic, right? Most of our organs in our body receive dual input, meaning that they receive input from the sympathetic and the parasympathetic nervous system. So we have our sympathetic nervous system coming in and innervating the heart, and we have our parasympathetic nervous system coming in and innervating the heart. Why is this advantageous?
Because you can control the homeostasis with it. You can either get excited or have it relax.
And more specifically, it provides precise control. Again, think of this in terms of your house. In your house, do you just have an air conditioner? That is certainly all you've been using for the past 6 months, or at least 3 months, right? But what happens when December rolls around? Is that air conditioner going to do you any good?
No.
No. I think I used the example in this. I usually use the example of this in my class. If you think back to the second day of class when we were talking about homeostasis, we were talking about that concept of a dynamic stability, where we have a narrow range of optimal conditions that we need to stay within. And so if you need to go above it, you need to bring it down. If you go below it, you need to bring it up. And temperature, again, was one of those examples. And as I mentioned, my sister, when she was pregnant, the temperature of her house that she was comfortable at was 73 degrees. If the house went above 73 degrees, she was sweating profusely. If it went below 73 degrees, right, it was, she was shivering uncontrollably. And luckily, she had one of those smart thermostats where she could set her range for 73 degrees. And so you'd be sitting there in the house with her, and the air conditioner would go on, and then 5 minutes later, the heater would go on, right? Because she wanted to be right at 73 degrees. And to do that, you have to have these opposing inputs. And that's what we have here. Obviously, what kind of an effect does the sympathetic nervous system have on the heart? Is it excitatory or inhibitory?
Excitatory. Excited.
Excellent. And the parasympathetic on the heart?
Inhibitory.
Now, does that mean that the sympathetic is always excitatory and the parasympathetic is always inhibitory?
No.
No. What happens if instead, and I don't have a lot of space here, but I can sneak it in. What happens if instead we have a stomach? Guess what? The stomach also has dual innervation, gets input from the sympathetic nervous system, and gets input from the parasympathetic nervous system. And what is the effect that the parasympathetic nervous system has on the stomach?
Excitatory.
Yeah, it is, after all, rest and digest, right? And if you need to run from a bear with an axe or fight a bear with an axe, is breaking down that cheeseburger that you had for breakfast one of your top priorities?
No.
No.
No, so notice it is inhibitory. So notice it isn't that one's always excitatory and one's always inhibitory. It's that they're always the opposite of each other. That is how we provide that precise control. And of course, not surprisingly, we have a fancy term for this when they have always the opposite effect. We say they're antagonistic. So we have these antagonistic inputs, opposite inputs, antagonistic inputs from the parasympathetic and sympathetic, and that gives us that precise control. And of course, we have a fancy name for this. We call this our autonomic tone. The same way we have muscle tone by having a little bit of a partial contraction going on to the muscle, we have autonomic tone, where coming into the heart, we always have sympathetic and parasympathetic inputs coming into the heart. Now, are the inputs always equal?
No.
No. When you're exercising, your sympathetic is going to be higher than your parasympathetic. When you're meditating, your parasympathetic is going to be higher than your sympathetic. So they're not always equal to each other, but there's always that autonomic tone coming in. Are we comfortable with that concept?
Yeah.
All right, because here's the problem with that. This word right here. While most of our effectors have that antagonistic input, have that dual innervation, what of course does most mean?
Not all.
Not all, absolutely. And so there are some exceptions to this autonomic tone, to this always antagonistic concept. The good news is most of them make sense. So the exceptions primarily get input just from the sympathetic nervous system. And like I said, some of these are pretty obvious. The adrenal gland only gets input from the sympathetic nervous system. Because what is the job of the adrenal gland?
Produce adrenaline.
Yeah. And when do you need adrenaline?
Flight or fight.
Yeah, when you're in that stressful fight or flight situation. So there isn't any reason to have your parasympathetic nervous system going to the adrenal gland. It's that pot filled with that adrenaline to provide us that stress response. So when we're stressed, we want to pour that pot out. But the rest of the time, we don't need a general level of adrenaline in our blood. We just need it when we're stressed. So there's no reason to have the parasympathetic go to it. Another interesting thing is, again, our skin. So our sweat glands, our rector pili muscles, the blood vessels that's way too big, to our skin, to our skeletal muscle. Why is this all capital? So it doesn't matter. Even to our adipose. Heart and lungs. These are all things that, again, when does your hair stand up on end?
When you're scared.
When you're scared, so you're in a fight or flight type of stressful situation, or when else?
Cold.
Cold, when we want to stimulate the muscle, increase the energy use of the body to bring our temperature back up. While you're sitting here calmly in your seat, are you sweating? No, but if you get out and around the room 16 times, or you have to fight that bear with an axe, you're going to get a little sweaty. Or when you're thinking about having to stand up and give that speech that's worth 50% of your grade, you can get quite sweaty during that type of a situation as well. Notice again, there's this pesky word, most. Most blood vessels receive input from our sympathetic nervous system only. When we're in that stressful situation, we have to redirect the blood where we need it to go. To the skeletal muscle, to the blood, to the nerves, to our heart, not to our nerves, to our heart, to our lungs. Right? We tend to get pale when we're stressed or scared because the blood vessels in our skin constrict to draw the blood away from that blood reservoir and send it to the heart and to the lungs and to places like that so that we can deal with that stressful situation. But there's that pesky word most again. There are some blood vessels that are innervated. There's a few blood vessels that are innervated by the parasympathetic. Any idea which ones those might be? It's another one of those cases where I know you know, even if you don't know that you know.
There's just the blood vessels go into your organs, your visceral organs.
Nope, not the ones to the visceral organs, although that's a really good guess. Well, so again, as I mentioned, I know you know, even though you don't know that you know. Because now that things are allegedly opening back up, although Delta and the number of unvaccinated people seem to be affecting that, but assuming that you and your others are vaccinated, you can now once again go out on that all-important third date, right? Third date on Zoom, not that big of a deal. But when you take that cute boy out for that third date and you buy him that really, really expensive meal and take him to that really, really expensive show at the, the opera or something along those lines, when you're dishing out that kind of money on a boy on a third date, you expect something in return. And he knows you expect something in return. And if living up to your expectations becomes a little bit too stressful for him, what can become the problem for that young man?
He'll dump you.
He starts sweating.
He could get sweaty, that could be part of it, but he also might have some trouble rising to the occasion, right? It turns out the blood vessels associated with the erectile tissue, that means the penis in males, that means the clitoris and the labia in females, those blood vessels associated with the erectile tissue are controlled by the parasympathetic nervous system. Which again, if you think about it, makes some sense. If that bear with an axe comes into the room, procreating probably isn't the first thing that comes to mind, right? And the bear with the axe might be a little weirded out if you get a hard on while you're trying to fight him. So wanting to reproduce, wanting to have that propagation of the species is a parasympathetic response. So if you're too nervous, right, that nervousness can affect performance. So do what I do with my boys, apply them with alcohol, and then they're much more less inhibited, and you can get what you need from them. All right. So like I said, most blood vessels, except for those associated with reproduction, the erectile tissue, again, penis, clitoris, labia, those are controlled by the parasympathetic nervous system, but all our other blood vessels are sympathetic only. Again, I think most of this, and especially when we think about the physiology of the autonomic nervous system, I think most of this stuff is fairly obvious. Here's the one that I think is somewhat confusing. The kidneys. What is the function of the kidneys?
To filter our waste out of our bodies.
Excellent. Waste out of our bodies, and primarily they take the waste out of what? You're right, out of the bodies, but where do they get, where are they actually taking the waste out of?
The blood.
Out of blood. Excellent. Right. Now, does that sound like the kind of process that is going to be controlled by your fight or flight sympathetic nervous system, or is that more of a housekeeping type of activity for your parasympathetic?
Parasympathetic.
Yeah, it absolutely sounds like a parasympathetic type of function. So it doesn't on the surface make sense that your kidneys should be innervated primarily by the sympathetic nervous system, because this is a housekeeping type of activity. But here's the catch. While you're sitting here calmly in your chair, about 25% of your blood is passing through your kidneys every minute. There is a huge amount of blood going to your kidneys. And when we have that huge amount of blood, let's write this out. A huge amount of blood goes to your kidneys. And obviously, the more volume that you have gives you a large pressure. And as we know, pressure makes the world go round. And so that high pressure allows the kidneys to work. Do their job effectively. Now, if you're sitting here at rest and 25% of your blood is going to your kidneys, do you really need your parasympathetic nervous system to tell your kidneys what to do?
No.
No, they're pretty much able to do it on their own. However, When you're active, when you stand up and you're running around the room 16 times, is 25% of your blood still going to your kidneys?
No.
No, and it doesn't even have to be you running around the room. You've got a 15-minute break in this class, so you need to walk downstairs, make a cup of coffee, grab a donut, and walk back up the stairs to your room. When you get up and walk down the stairs, now I'm not even talking about up the stairs, but as you're walking down the stairs to get your cup of coffee, is 25% of your blood still going to your kidneys?
No.
No. And so that's a problem. When active, there is much less blood going to your kidneys. And that's a problem. Less blood means less pressure. And with less blood pressure, the kidneys are not as effective. Now, how important is it that your kidneys filter your blood?
Very.
Yeah, if I rip the kidneys out of your body, how long would you survive for?
Not long.
Not very long, right? If your kidneys stop functioning, you're doomed. You'll last a couple days at best. Yes, with things like dialysis, they can prolong that, but dialysis really isn't a cure. It's a stopgap, basically, to allow you the opportunity to hopefully get a kidney transplant, because even as sophisticated and as improved as those dialysis machines have gotten, they're not able to take the place of what the kidney can do. So if every time you get up and walk 10 steps, blood starts going to other parts of your body, you don't want your kidney function to decrease. So what our sympathetic nervous system is able to do, our sympathetic nervous system is able to increase the pressure in the kidneys, even when there's less blood. And so if our sympathetic nervous system keeps the pressure up, the kidneys can function normally, even though they have less blood. So like I said, on the surface, it sounds counterintuitive that our kidneys would just receive input from the sympathetic nervous system. But it's because at rest, they're so efficient at what they're doing, the parasympathetic nervous system doesn't tell them what to do. But when we're active, the kidneys need help. And so our sympathetic nervous system helps the kidneys when we're active. So our kidney function doesn't go down. Now, is it going to be perfect? No. If you really are running or fighting that bear with an axe, you're going to get a decrease in kidney function, right? But if you're fighting with that bear with an axe for your life, that fight's probably not going to last very long, or you sprinting away from it's not going to last very long. And if you're sprinting from a bear with an axe to try to survive, is a slight decrease in kidney function your biggest concern at that period of time?
No.
No, because typically these stressful situations only last a very short period of time. And that's one of the major problems with stress. Because while in ancient times it was bears with axes that caused us to have that stress response, because we felt we were in a life and death situation. Now the stressors that make us feel like we're in a life and death situation is that 10-page paper you have due on Friday, or that 100-point cumulative exam that you have on Friday, or those constant deadlines at work. If you have an incredibly stressful job where you're constantly in this state of stress, that is going to decrease blood flow to the kidneys, right? This constant input can actually cause damage. Chronic stress can be very damaging to the kidneys. And as we talked about, your kidneys are really, really vitally important in their function. So it's one of those things we have to be really careful of. Unfortunately, the things that stress us these days are not the types of things that stressed us before. And so chronic stress becomes a much more serious concern, and it can have a lot of long-term effects and implications on the human body. which is why I told you not to take summer school. All right, questions on that?
Yeah, I got a couple. So when it comes to the sweat glands, and it's talking the sympathetic only. So like if you're sitting there and you're hot and you're sweating, is that just like a different setup going on? Because I mean, you're not really stressed, or is that your body just doing its thing?
So again, if you exercised, you're active, right? And as you're active, your sweat glands are going to sweat to help to lower that body temperature, right? At the same token, if you're in a sauna, that heat, as we talked about, an increase in body temperature increases heart rate, right? Increases our sympathetic activation. So even though you're not physically active, your body is responding to external stimuli, and that's going to increase our sympathetic activation, which will then cause our sweat glands to become active to produce the sweat to try to bring our body temperature back down.
Okay, so that's just based
Normally,
upon the temperature.
yeah, normally you're just, yeah, normally you're not just sitting there and sweating just from the act of breathing, right? It's either you just finished doing some type of activity, or like we talked about, maybe you're stressed because you have to give that presentation, or maybe because your air conditioner is not working and it's 85 degrees in your house, right? Any of those types of things could cause you to sweat, and all of it is caused by that increase in sympathetic activation. Does that make sense?
Yes, it does. And then the
You
other
said you had
one
a
was,
couple questions. Yeah, go ahead.
yeah, the erector pili muscles. Like, yes, I understand you're scared, you're excited, something like that. Your hairs will stand on end. But like for me, I'm musically inclined. If I hear like a really good music or like some really good singing, my hairs will stand up. How exactly, if there's an answer to that, I don't know.
It is a great question. There are people who they've noticed who are, I'm trying to think of this isn't the right term for it, but that are more in tuned to music, more likely to be physiologically affected by music. And yeah, in that case, the music is stimulating a sympathetic response to you. And so that can cause your hairs to stand up on end. So it's still going to be the sympathetic nervous system. Remember, as we talked about, this is just a motor output. So any type of sensory information coming in, whether it is temperature from the skin, whether it is seeing that bear with an axe, or whether it's hearing, some really moving music, or watching some really, really emotional Hallmark video. I mean, a commercial, like a Hallmark card video or a commercial or something along those lines. Those types of things, heck, if you really sat here in a quiet environment and really thought about it, can you stimulate these types of responses? If you really sat for half an hour just with your eyes closed and tried to envision exactly what it would be like to have to stand up and give a presentation in a class that was worth 50% of your grade, and you had to do it in your underwear. If you really sat there and thought about that, could you get yourself to get sweaty, get your heart to start racing and start thinking about it from just thinking about something like that? Yeah, absolutely. So it doesn't even have to be an outside stimulus. Just the cognitive thinking of it can have these types of influences and effect on us, right? If you really sat there for 15 minutes and thought about the best meal that you ever had at that fancy restaurant, and thought about the smell and the aroma of whatever the meal was, or the taste of it, could you get your salivary glands to go? Absolutely. So external stimuli, internal stimuli, cognitive thought, all of these things have an ability to influence this motor output, right? Remember, this is just a motor output. Different stimuli can cause it, different processing of the nervous system can cause it. So whatever the cause is may change, but the effect is the same. We have this autonomic output that stimulates the sweat glands to sweat due to any number of external or internal stimuli. All right. Excellent. All right. Any other questions on this? Perfect. Where are we on time? Okay, excellent. So we've got this concept now where we understand that there are the two branches of the peripheral nervous system, our sympathetic, pardon me, of our autonomic nervous system. And that they are the sympathetic and parasympathetic. And not surprisingly, they are going to be different in their function and their anatomy. And so that's what we need to do next. What we need to do next is start comparing our sympathetic and our parasympathetic nervous systems, both anatomically and physiologically. And I think the physiological is easier. I think this is the one that we're more comfortable with, we're more familiar with. So this is where we will start. But even though it's easier, I think it's still, we're at a good stopping point for our first break. So let's go ahead and take our first break. We will take a 15-minute break as always. So it looks like that means we'll return at 9:42. And I will start the recording at that time. And so unless there's any questions, I will see you guys in 15 minutes. Any questions? All right, see you guys in 15 minutes. All righty. Back at it. So, as I mentioned, I think the physiological effects of the autonomic nervous system are the ones that we are most familiar, most comfortable, most obvious. So give me some examples. What are some of the examples of the way our sympathetic nervous system affects our body physiologically?
Heart rate increase.
Yeah. And not just heart rate increases, but when you're scared, not only does your heart beat faster, but it also beats stronger as well. You can feel it beating in your chest. So the strength of the contraction increases as well. So you can feel it literally pounding in your chest. Let's make that a little smaller. Excellent. All right. Give me something else.
Pupils dilate.
Excellent. Bear with axes are incredibly tricky organisms. So we want to open up our eyes, be able to take in as much light as possible so we can differentiate our environment and try to find where those sneaky camouflage bear with axes are. What else?
Airway Styling.
Absolutely. We're going to relax the smooth muscle of our airways so we get more air in, meaning more oxygen in, because if we're in that fight or flight situation, we are going to be using energy. And as we know, we need oxygen to efficiently break down those sugars so that we can get all the ATP we can. What else?
Blood vessels constrict non-essential organs.
Excellent. I like that. We have blood vessel effects. And as you've hinted at, there are two different effects that we're going to have on blood vessels. Blood vessels to non-essential organs, they are going to constrict. So as we talked about, this includes things like the skin, like our digestive system. Right. These are areas where we talked about our skin is a blood reservoir while you're at rest. So it constricts. We tend to get more pale when we're stressed, when we're scared, because the blood moves away from our skin. Our digestive system, it's one of the reasons why you're not supposed to swim for a half an hour after you've eaten, because you've had that nice big cheeseburger for breakfast and then you immediately hit the pool. As you're vigorously swimming back and forth in the pool, we decrease blood flow to the digestive system. You're not able to completely digest and break down that food, and that can lead to intestinal cramping as a result of it. However, what about two essential organs? What do we want to do to the blood vessels there?
Dilate.
Excellent. And let's be more specific. What do we mean by essential organs? What are some of the structures or regions of the body that there's going to be an increase in blood flow?
Heart, lung, kidney, brain.
Remember, not kidney. Remember, kidney is one of those areas where the blood vessels constrict. Less blood goes to the kidney, but remember what we do is the sympathetic nervous system helps to keep the pressure up. So even though less blood's going there, we're going to try to keep the pressure up there. So we do keep a high pressure, but not because we're dilating the blood vessels there. Less blood's going there. So we want to make sure that they are keep that pressure high. So heart, lungs, where else? I'm sorry,
Brain.
say again?
Liver.
Liver. Why do we want to increase blood flow to the liver? You're 100% absolutely correct, but do we know why?
Break down glucose for energy.
Exactly. Absolutely. Remember, one of the things we need to do, as we talked about, we dilate those airways so that we can get more oxygen into our body because we need to break down fuel. And so we need that fuel. We're going to go to the liver to get glucose. We are also going to go to our adipose tissue to get fuel so that we can break it down. Right? This is one of the problems with stress. Again, remember, stress used to be that you had that fight or flight situation. You were releasing all this fuel into our blood because you either had to fight or you had to run. Right? I was glorious before I had kids. Right? But then I had kids. Now I'm in a constant state of stress. Right? And when you're stressed about your kids, your body's physiologically releasing all of this fuel into our blood. But if you're not running a marathon, if you're not fighting a bear with an axe, what happens to all that fuel? Does it get used? Yeah, it doesn't get used, and so it gets stored. It gets stored as adipose. It gets stored as fat, right? I had hair. I had a six-pack. I was, like I said, I was glorious before I have kids. Now I'm falling to pieces. Excellent. Heart, lungs, liver, adipose. I can think of one more really obvious place we want to send a lot of blood.
Skeletal muscle.
Muscles.
There we go. Skeletal muscle. Perfect. So these are the places we want to send blood to, and we want to shift it away from those non-essential areas. Excellent. What else?
Release of glucose from liver and adipocytes.
I'm sorry, say that again.
Release of glucose from liver and adipocytes.
Absolutely. As we said, we want to not only increase blood flow to those areas, but then we want them to release their fuel so that we have that readily available to be able to break down. Awesome. Anything else?
Decrease digestive activity.
Notice, as we talked about, obviously that includes stuff like the stomach and the intestines. But remember also that involves, for instance, our salivary glands. So, for instance, our mouth gets really, really dry. But what isn't dry when you're stressed out?
Skin.
Yeah. So while things like our salivary gland or our digestive glands decrease in function, our sweat glands become highly active. So your palms get cold and clammy. Cold because we're constricting the blood flow, so less blood's going to it, so it gets cold, and it gets all sweaty, so it's evaporating, getting cold from that, and so it's cold and clammy. But like we said, your mouth is dry as a bone. Anything else? I think that's pretty good. And notice also the other important thing to remember about our sympathetic nervous system is notice this is giving us a big global effect. Notice our pupils don't dilate, and 5 minutes after that, our airways dilate. And 6 minutes after that, blood vessels to our stomach decrease. And 5 minutes after that, we start producing sweat. And 3 minutes after that, our heart starts beating. We have big global effects, and this is occurring all at once. And again, one of the reasons is, as we talked about with our sympathetic nervous system, the other thing that is affected by the sympathetic nervous system is the activation of the adrenal gland. And of course, what does our adrenal gland do?
Produce adrenaline.
Yeah, adrenaline, releases adrenaline or epinephrine into the blood. And as we mentioned, once it gets into the blood, where does it go?
Everywhere else.
Everywhere.
Everywhere, giving us that big global effect. And again, you may not have thought of it in these terms, but this is our last big week of class. So you could be sitting there quietly at the kitchen table studying at 2 o'clock in the morning. And then bang, there's this loud noise outside, right? You think it's gunfires going off or somebody else, horrible things going on. And then in about a second, you realize that it was just somebody's car backfiring. 3 seconds after it occurred, you know you're safe. But does your heart rate instantly go back to resting? No, because that instant of stress caused that adrenaline to be released. And that adrenaline is an hormone. It's a long-lasting chemical signal. So it gets into your blood, causing this stress response where your heart's beating faster and stronger, right? And all these other effects that we talked about are going on. And it lasts for 15, 20 minutes before your heart rate finally goes back to resting again. So we have this big, huge global response. And I think we hit all of it, but again, it's about using energy, dealing with that stress response. So we need to use and mobilize energy. So we talked about our pupils dilate, heart rate, force of contraction. Oh, we didn't mention blood pressure. Obviously, if the heart's beating faster and stronger, blood pressure is going to increase. Airways dilate, blood vessels to non-essential organs constrict, heart, lungs, skeletal muscle, liver, adipose dilate, release that glucose level into our blood, getting that fuel readily available, decrease digestive function, right? Sweating, which isn't on this list, but we talked about as well. Excellent. And here's the awesome thing. We know our sympathetic and parasympathetic are antagonistic with each other. So whatever the sympathetic does, the parasympathetic essentially does the opposite. If the sympathetic dilates the pupils, what does the parasympathetic do?
Constrict.
There you go, constricts the pupils. If the sympathetic increases heart rate, what does the parasympathetic do?
Decrease.
Right. If our sympathetic decreases digestive function, what does our parasympathetic do?
Increase.
Yeah. So again, that is, once you learn one, you know the other. Although we did talk about how that rest and digest wasn't necessarily the best mnemonic to help us to remember the function of the parasympathetic. Remember, this is about our housekeeping type of activities, our storing of energy. And there are a couple good ways to remember what these things are. One of them is the acronym SLUD. SLUD stands for salivation, lacrimation, urination, digestion, and defecation. Salivation is, of course, production of saliva. What's lacrimation?
Production of tears.
Tears. So producing saliva, producing tears, producing urine, defecating, digestive function. All of these are major physiological functions that are controlled by the parasympathetic nervous system. Another important mnemonic for our parasympathetic nervous system is to remember the big three. Remember, I gave the example. I'm pretty sure I've given the example. I have a nurse who was a former student from mine in both 430 and 431. And as I mentioned in the 430 class that she took with me, they have a cabin up in Tahoe, and her boyfriend, not once, not twice, but 3 times, liked to roam around the countryside and eat random mushrooms that he would find on the hills. And 3 separate occasions, he ate the wrong types of mushrooms. And they had to call the EMTs to come and save his life. And what happens with those particular mushrooms that he was eating is they stimulate the parasympathetic nervous system. So he eats these mushrooms and his heart rate would decrease, his airways would decrease, and his pupils would become very, very constricted. Notice, as we also talked about, he would start producing tears, he'd start producing saliva. And how good of a combination is that? You've got a mouth filling up with saliva and a constricted airway. Is that a whole lot of fun?
No.
No. Also, confusingly, but we'll see why in a little bit, he'd also start sweating profusely as a result of that, right? This is not a fun condition to be in. And yet those mushrooms must be really, really good if he's willing to risk it to be able to eat them. So we have these 3 big decreases from that parasympathetic input.
Why the tears?
Why does it produce tears?
Mm.
That's a great question. Why we cry is one of those questions we don't fully understand as well. It is believed that it may be a way of helping to relieve stress. But if nothing else, we know tear production, aside from the emotional standpoint of it, play an important role in that housekeeping of the eye, washing away any dust and debris, maintaining the moisture of it. And so again, remember, our parasympathetic is kind of our housekeeping type of activities. And so definitely keeping your eyeballs clean is a housekeeping type of activity. And so it could be that. But then, yes, you're right. There is all the emotional components to it as well. Remember, as we talked about, there is that arousal. When we get to the reproductive system in 431, cute, simple mnemonic you'll be able to remember is point and shoot. Because when the reproductive system, the point, the arousal or erection, right, of penis, clitoris, and labia, is a parasympathetic reflex, and the shoot, the orgasm, is a sympathetic reflex. So both the parasympathetic and sympathetic nervous systems play a role in reproduction. And to remember which is which, you just have to remember point and shoot. All right. Now, remember one other big difference between these two. While our sympathetic is big global responses, notice that our parasympathetic is discrete local effects. All right. When you go to, right, because this weekend's your anniversary, so you take your loved one, again, assuming they're not vegetarian, to Ruth Chris for the for a nice fancy dinner, and they bring out that beautiful aged sirloin with that herb butter on it and all that. You may start salivating as a response, but do you cry when you see that steak?
If it's a tomahawk, yeah.
Okay, maybe if it's a tomahawk, or maybe when you get the bill at the end, you might cry a little bit. But even though these two glands are right next to each other, you're not salivating and crying at the same time. Right? Thankfully, when you're aroused, you don't defecate at the same time. Right? So typically, these are local, discrete effects. Our sympathetic is big, huge, and global. whereas our parasympathetic is local and discrete. And we see that not only in the physiology, but we're really going to see it in the anatomy when we look at the anatomy of these. Again, the good news is I think all of this shouldn't be surprising information for you. I think most of us kind of understand the physiology of fight and flight and rest and digest, and we're familiar with that. Your book's got a nice table that goes through those that you can look at that summarizes it nicely. But like I said, I think the physiology is the easy part. Where things get a little trickier is in the anatomy. Now notice here, we see our basic autonomic pathway. We have a preganglionic neuron whose cell body is located in the lateral gray horn. Although our artist hasn't done the best job of doing that. They really should have put it really up here more in the lateral gray horn, but they gave us a tiny lateral gray horn, so I guess that's good enough. It has a myelinated axon that goes to the ganglion, releases acetylcholine on our postganglionic neuron, which is unmyelinated, which is going to either release acetylcholine or norepinephrine. So we have this, we already know, as our basic autonomic pathway. But not surprisingly, there are going to be some differences between the sympathetic and the parasympathetic for their pathways. Now, they're both autonomic, so they're both going to have two motor neurons, a preganglionic neuron, and that one is the one that goes from, this is going to be too big, central nervous system to ganglion, and a postganglionic neuron, which goes from ganglion to effector. So those are going to be the same for both of them. Ugh. I spelled two wrong as well. Preganglionic's myelinated, postganglionic's unmyelinated. So again, they're going to share those characteristics, but there are going to be some differences. One difference is going to be where they're located, what their axons are forming. The second is going to be a branching, and the third is going to be the neurotransmitters used. So just like before, let's draw these. Again, we'll draw a line down the center so we can compare the two. And let's start with our sympathetic pathway. We know it has to start in the central nervous system. We know it needs to get to its effectors. And we know those effectors are going to be, I'm not going to bother drawing them this time, smooth muscle, cardiac muscle, glands. And we know our parasympathetic needs to do the same thing. Excellent. We know let's start with our sympathetic. we know that there are 2 neurons in this pathway. The first neuron is our preganglionic. And we know some things that are true for both the parasympathetic and the sympathetic. We know that they start in the central nervous system and they go to the ganglion. We know that it has a myelinated axon. And we know it releases acetylcholine and it is excitatory.
Multipolar.
Oh, excellent. And we know one more thing about it as well. Excellent. So these are things we absolutely positively know about our preganglionic neuron. But the other thing we know is about its ganglia. Let's try that again. Sympathetic ganglia are located close to the central nervous system. So since it's closer to the central nervous system than it is to the effector, oops, no, hold on, that to be black. I want this to be red. So we know it's a multipolar neuron. We know its axon is going to leave out the ventral root to go to the ganglion. And if the sympathetic ganglia are close to the central nervous system, what is the relative length of our preganglionic neurons' axons? Are they going to be relatively long or relatively short?
Relatively short.
So it's going to have a relatively short axon because it is so close. And the other thing, I guess I can put this over here, I have a little room, is remember we have to make with our sympathetic a big, global, large effect. As we know, axons can produce as many as 10,000 synapses. Do you think this one's going to form a lot of synapses or just a few synapses?
A lot.
Lots. And so to do that, its axon terminals have to branch elaborately. So this axon is going to come out. It is going to branch elaborately and form many, 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 many synapses. Of course, we can't forget to myelinate our axon. Remind us of that. Excellent. And so it branches elaborately, forms many synapses. And has a relatively short axon. Now, as we know, it always releases acetylcholine. And that acetylcholine is always excitatory. On our postganglionic neuron. Let's talk about things we know about our postganglionic neuron. Oops, hold on. Don't want that all caps. Again, we know it starts in the ganglion and it's going to go to the effector. What is its structural classification?
Multipolar.
What is the
Unmyelinated.
state of its there? Unmyelinated axon. Excellent. Axon. And when it comes to the neurotransmitter, most release norepinephrine. Of course, that does. Oh, wait, so let's really do this. So that's all things we know. Most release norepinephrine, but there are some that release acetylcholine. Again, we know the effect can be excitatory or inhibitory, depending on neurotransmitter and the effector. So that's still going to be true for this, for all of them. But here in this case, most produce norepinephrine, some release acetylcholine. And if the ganglia are close to the central nervous system, what is the relative length of our postganglionic neuron?
Relatively long.
It's going to be relatively long. And once again, for a big global effect, are we going to want to just have one synapse or many synapses?
Many.
So to do that, we have to branch elaborately. So its unmyelinated axon comes up, again, forms many, many branches. And again, most release norepinephrine, and some are going to release acetylcholine. All right. Questions on that?
I might be jumping the gun, but is there a reason why it's myelinated before and unmyelinated after?
Why is one of those questions that, as we've talked about, is challenging in anatomy and physiology, right? Because again, as we mentioned, the engineer who designed all this did not leave us her footnotes. We can often infer why things might be set up that way, because often there is some intuitive or obvious logic why something would be more beneficial. I have not heard of a good explanation as why one would be myelinated and one would not be myelinated, so I don't have a good answer for you for that. I don't know why.
Okay. I was wondering.
Yeah, no, again, and you're right. Sometimes we can kind of figure out why, because like I said, there might be an obvious rationale, or there might be something where it's intuitively, you know, smart that it's set up that way, but I don't have a good answer for you for this one. All righty, let's do the same thing in the parasympathetic nervous system. For our parasympathetic nervous system, so this over here, and we'll do this time. We know that, again, we are going to start with a preganglionic neuron. And just like we saw in the sympathetic, it is going to start in the central nervous system, and it's going to go to the ganglion. It is multipolar, and it has a myelinated axon. Right? That information is the same for both of them. Also, it is going to always release acetylcholine, and it is always excitatory. So again, these are true for both our parasympathetic and sympathetic preganglionic neurons. However, one of our big differences is the location of the parasympathetic ganglia. Parasympathetic ganglia are located either right next to, or in some cases, they can actually be in the walls of the organs, of the effectors. Many of the ganglia, for instance, the parasympathetic ganglia of the heart, of the stomach, of the small intestine, those are actually located within the walls of those organs. Others, it's right outside. So obviously, these are much, much closer to the effectors than they are to the central nervous system. And so, of course, what does that do to the axon length of our parasympathetic nervous system? What's the axon length of our preganglionic neuron if it has to go from the central nervous system almost all the way to the effector?
Very long.
Yeah, it's going to be relatively longer. And while our sympathetic wants to big global responses, remember our parasympathetic, we want small discrete responses. So are we going to want to have a lot of synapses or just a few synapses?
Few.
And if we only want a few synapses, are we going to branch elaborately or are we going to stay discrete?
Discrete.
Excellent. So we have our multipolar preganglionic neuron located in the lateral gray horn, for instance, coming out the ventral root, going all the way to our ganglion, where it is going to very discreetly branch and only form a few synapses. And let's not forget that this is myelinated along the way. Whereas if our postganglionic which starts in the ganglion and goes to the effector. It is also multipolar. It is unmyelinated. Again, those things are still true. Oh, we forgot one more thing. We know that these always release acetylcholine, and it is always excitatory. We have our multipolar neuron with its axon that is unmyelinated. But as we've talked about, if this ganglion is right next to or actually inside the wall of the effector, what is the relative length of this axon?
Short.
And again, we want discrete innervation. So once again, it is going to form few synapses and it is going to have discrete branching. But there's one other big difference between our sympathetic and parasympathetic. Here in our parasympathetic, all postganglionic neurons release acetylcholine. But as we know, it can be both excitatory or inhibitory, depending on the effector. Notice both pathways use two neurons to get to their destination, starting in the central nervous system, going to the effector, some having positive, some having negative, depending on what the effectors are. But there are also some big differences. Sympathetic, big global branching, many, many synapses for a big global response. Parasympathetic, very discrete branching, very few synapses, very discrete local effect. Sympathetic nervous system's ganglion are very close to the central nervous system. So their preganglionic axons are relatively short, whereas their postganglionic axons are relatively long. And it's the opposite for the parasympathetic, because their ganglia are so close to the effectors, sometimes even inside the effectors, that the preganglionic is much, much longer and the postganglionic is much shorter. And then they also differ in the neurotransmitter they release. At least from the postganglionic neurons. Notice all preganglionic neurons all release acetylcholine and it's always excitatory. So where they are different is what that postganglionic neuron is doing. For the parasympathetic, all of them release acetylcholine, whereas for the sympathetic, most release norepinephrine, but a few will release acetylcholine. Again, I've done an amazing job of drawing this myself, but if we look at the pretty pictures, notice here we see the sympathetic pathway. Ganglion is closer to the central nervous system, so our preganglionic axon is relatively short and our post is longer. But notice that both of them branch elaborately, forming many, many synapses. Notice while our preganglionic neuron always releases acetylcholine, most of the postganglionic release norepinephrine, though there are a few that will release acetylcholine. Conversely, when we look at the parasympathetic, again, notice the ganglia is much closer to the effectors. So our preganglionic neuron has the much longer axon and our post is much shorter. But both our pre and postganglionic all release acetylcholine. And very discrete branching, very few synapses, giving us a small local effect. Notice if we come back to this picture from your textbook, we now see that they were actually showing us this. Here in our autonomic, it is showing how our sympathetic ganglia are closer to the central nervous system, whereas the ganglia and the parasympathetic are closer to the effectors. All postganglionic neurons release acetylcholine, whereas in our sympathetic, most release norepinephrine. And when we talk about big global effects, the other thing that our sympathetic nervous system does is cause our adrenal gland to release epinephrine and norepinephrine into the blood, which not only gives us a big global response, but a longer lasting big global response. So again, we see here how they don't notice the difference in branching. This one branches much, much more. This one branches much less. But other than that, they do a nice job of showing us that difference. Questions on that? It only gets worse from here. So if this isn't making sense, ask now, because it's about to get a whole lot worse. I warned you. Notice we have this scary looking octopus type structure. This and many of the illustrations like it are very intimidating when you first look at them. However, one of the things that I want to point out is as you get more and more comfortable, more and more familiar with this material, you'll see that it makes more and more sense. Notice some of the things that we already know. For instance, the smooth muscle of the eye receives dual innervation. Both the sympathetic and the parasympathetic nervous system innervate it with different effects. Sympathetic's going to dilate the pupil, and parasympathetic is going to constrict it. And notice we can see that it takes 2 neurons to get there. Neuron 1 starts in the central nervous system, synapses on a ganglia right next to the eyeball, where our second postganglionic neuron has a much shorter axon to innervate the eye. Notice in our sympathetic pathway, its synapses are first preganglionic neuron, synapses on a ganglion, much closer to the central nervous system. So our postganglionic second neuron's axon is much, much longer to get to the eye. Notice whereas there was just one path to the eye because it was very discrete, notice there's a lot of innervation. So I could get driving directions from almost any of these locations and get up here to be able to get to the eye. Big, huge global effects. And notice, well, there's eye on this side and eye on this side and salivary gland on this side. I don't see skin over here on the parasympathetic. I don't see adrenal gland over here on the parasympathetic. Why not? Does the parasympathetic nervous system innervate the skin? Does it innervate the adrenal gland?
No.
No. So not surprisingly, they're not effectors on the left side of this because there is no pathway from the parasympathetic nervous system to the adrenal gland or to the skin. So notice, even with the limited bit of information we have already, we can start to tease out some of what's going on this octopus. Now, obviously, there's a whole lot more going on here than we've had a chance to talk about. And that's one of the other big things we're going to be able to see. Notice both our sympathetic and parasympathetic are going to start in the central nervous system. But do they both start in the same parts of the central nervous system?
No.
No. Notice that our sympathetic basically starts in the spinal cord from T1 all the way down to about L2 or L3. So notice anatomically, the sympathetic nervous system is sometimes referred to as the thoracolumbar branch, because where does it come out of the central nervous system?
Thoracic and lumbar.
From the thoracic and the lumbar region of the spinal cord. Notice our parasympathetic comes out of the brain stem, like where our cranial nerves are located, and the sacral portion of the spinal cord. So not surprisingly, anatomically, it is referred to as the craniosacral branch. So notice they are anatomically given names by where they start in the central nervous system. Another way that we tell the difference between the sympathetic and the parasympathetic. Questions on that? I know it doesn't make this any less scary looking of an octopus, but we're going to continue to work through it over today, the rest of today and tomorrow, and see if we can make more sense of it. And the good news is we'll dip our feet into the kiddie end of the pool first. Not surprisingly, as you look at this, as we know, our parasympathetic is more discrete, whereas our sympathetic is more global and therefore more messy. Our parasympathetic is more discrete, and so it's a little bit easier to follow. And so that is where we're going to start. We're going to start first by exploring these pathways in the parasympathetic, or remember what we call the craniosacral branch of the autonomic nervous system. Now, it's a teeny bit early, but I want to come at this information fresh. So let's go ahead and I'll leave this on so it can be scary and intimidating while we're looking at it, but we're going to go ahead and take our second break right here. We will again take a 15-minute break, so it looks like that's coming back at 10:50, and at 10:50 we will restart, and I want to make sure that I start the recording, because you are going to need to look at this and listen to this again, because it's scary and intimidating the first 5 times you look at it. But the good news is by the time you look at it a 6th time, it's going to make total sense. And I also know, being the smart, sophisticated students that you guys are, that you guys started this and already looked at it twice over the weekend. So I'm sure this will be the 3rd time we're going over this material, so it'll hopefully start to make some sense now. All right, questions on any of that? All right, I will see you in 15 minutes. All righty. So our goal now is to understand the specific parasympathetic pathways. All right, and that's what we're dealing with here. Now notice we have this scary looking half an octopus looking picture that shows this, but clearly you're not going to draw this, nor do you need to draw this. But we can use it as an important component to be able to figure out the things that we need to know. Clearly, to understand the specific parasympathetic pathways, we need to know certain pieces of information. One of the things we need to know is clearly where the preganglionic cell bodies are located. We need to know what structures their axons form. All right, so when they leave the central nervous system, what is it that they become? we need to know and identify where, or let's say names of the specific ganglia, where the postganglionic cell body is located, and then ultimately we need to know the path to a specific effector. So these are the things that an illustration like this shows us. But we can also do this like on a chart. So let's make it a little easier by doing this on a chart. For that, we'll get the whiteboard, clear the images that is here already. We need to know our specific parasympathetic pathways. And as we mentioned, there are basically 4 pieces of information we need to know. We need to know where it starts in the central nervous system. And again, by that, we can think of this as where the preganglionic cell body is located. We need to know the structures formed by the I'm just going to go preganglionic axons. We need to know the parasympathetic ganglia. We need to know the effector. I can't spell effector correctly. And then kind of along with this, our postganglionic axon, what it forms. Now, remember this branch of the nervous system, parasympathetic, is known as the craniosacral branch. Because when it comes to where in the central nervous system our cell bodies are located, there's basically 2 main locations. They're in the brainstem, right, associated with our cranial nerves, and they are in the sacral portion, or let's be more specific. In the lateral gray horn of the sacral spinal cord. And I guess we should have been more specific up here as well. And that's too big. There. Nuclei and the brainstem. All right, so those are the 2 possible starting points for our parasympathetic pathway. And if we go back to the illustration here, notice that the ganglia located here in nuclei in our brainstem happen to have axons that come out to help form not one, not two, not three, but 4 cranial nerves. So what are the 4 cranial nerves that are formed by axons in our parasympathetic pathways?
Like the number or the name?
Well, let's do number first, and then once you give me the number, because that you should be able to read right off of the board here, you can then give me the name. So give me one of the numbers.
3.
3, excellent. And what was the name of 3 again?
Oculomotor.
Motor.
Excellent. Give me another one.
7.
7. And what was that name?
Facial.
Excellent. Give me another.
9.
What was its name?
Glossopharyngeal.
Glossopharyngeal. And what is the last one?
10.
And what is its name?
Vegas.
Excellent. So notice if we go back to our whiteboard, we can see that some of the structures that are going to be formed by our preganglionic axons are going to be some cranial nerves. And which specific ones? Well, cranial nerve 3, which, as you guys told me, was oculomotor. Excellent. Cranial nerve 7, which was facial. Cranial nerve 9, also pharyngeal. And cranial nerve 10, which is vagus. Excellent. Now, notice we didn't necessarily learn all the names of all the nuclei. We learned some of them, but we didn't learn all of them. And so we can simplify this a little bit. Not surprisingly, the cell bodies that whose axons form cranial nerve 3, we can just say that they start in the ganglion for cranial nerve 3. So there's a ganglion for cranial nerve 3, and its axons form cranial nerve 3, the ocular motor. There is a ganglion in the brainstem for cranial nerve 7, and its axons form cranial nerve 7. Ganglion for cranial nerve 9, and of course its axons form cranial nerve 9. There we go. And lastly, there's a ganglion for cranial nerve 10, and its axons form cranial nerve 10. So we don't need to learn the names of any new specific ganglia. Do they have names? Yes, absolutely. Do we care what their names are? No, we don't care. We can just call them what they are. The ganglia of cranial nerve 3 forms, its axons forms cranial nerve 3. Now, we also know that these are going to need to synapse on some specific ganglion. And as it turns out, and we'll go back and we'll cheat and we'll look at the picture of this in a minute, but it turns out that in the entire parasympathetic nervous system, there are only 4 named parasympathetic ganglia. As we have learned, anatomists love to name everything. But it turns out even our anatomists start to hesitate a little bit when we talk about these tiny little ganglia that are located right next to, or in many cases, inside the organ. And so if they're located inside the organ, there's not even a point in giving them a name. In fact, and I'll write this down here at the bottom, but we'll move this in a moment. Most aren't given a name. They're just simply referred to as terminal ganglia, just a generic term. We see this if we go back to our octopus. Notice, for instance, if we look at the lungs or the heart or the liver or the stomach or the spleen or the pancreas or the bladder, or the uterus or the bladder or any of these structures, they're either in the organ or right next to the organ. There are only 4 ganglia that have names. And notice the only named ganglia, the 4 named ganglia, the only ones with names, are all found in the head. So let's follow our pathways. I'm going to go back to the whiteboard. Hopefully you guys have some way of either having your textbook or some of the handouts or some of these other illustrations next to you to help you, so that you could see, for instance, that the axons in cranial nerve 3, the ocular motor, go to one of our 4 named ganglia. And anyone catch what the name of the ganglia that is innervated and synapsed on by the ocular motor nerve might be?
Ciliary.
Very.
There you go. Ciliary. Oops, that doesn't need to be capitalized. It is the ciliary ganglion. Excellent. And what does it innervate? What is the effector that is innervated by cranial nerve 3?
Thigh.
And remember, we talked about this with our cranial nerves. Because this is autonomic, this would be the smooth muscle inside the eye. Remember, that would be what changes the focus of our lens or changes the amount of light that comes in, the iris or the lens. Excellent. Now, one last thing. These postganglionic axons, if they are right next to or actually inside of the organ, do you think they're going to have precise specific names?
No.
Probably
No.
not.
So they are just, for all of our pathways, this postganglionic axon is just referred to as a parasympathetic nerve. So really, anything as it leaves the ganglia, whether it's a terminal ganglia or a named ganglia, we just call it a parasympathetic nerve. So a parasympathetic nerve leaves the ciliary ganglia and goes to the eye. And in fact, I'm going to cheat. Of course, to cheat, I need to go back to a different lecture.
What does ciliary refer to?
The muscles that move the lens are called ciliary muscles. So they're actually a structure inside of the eyeball. Notice, if you remember when we were learning all the muscles that move the eye, and we were learning about cranial nerve 2, cranial nerve 3, and cranial nerve 6, notice here we actually talked about it. We didn't name it at the time, but we knew that some of the axons from cranial nerve 3 innervated a ganglion, which then innervated the smooth muscle in the eye. We mentioned that briefly, and lo and behold, we see now that that ganglion we briefly mentioned is indeed that ciliary ganglion. So here we actually see that first parasympathetic pathway. Axons leaving the brainstem, coming out cranial nerve 3, synapsing in the ciliary ganglia, where our postganglionic neurons will then innervate the eye to move our lens or to change our iris. And there you go. Just that simply, we have seen, identified, and talked about our first pathway. So for instance, on a lecture exam, could I ask you to identify or describe the pathway of how the parasympathetic nervous system would innervate the eyeball?
Yeah.
Yeah, I'm asking the question, so the obvious answer is yes, but yeah, absolutely. And notice now with that previous picture, we can actually even identify the ciliary ganglia because we've seen it. Excellent. And it works the same for the others as well, starting with cranial nerve 7. Again, hopefully you have the picture in front of you, because if you do, that's going to make this next question a lot easier for me to ask. What is the ganglion that is innervated by the 7th cranial nerve?
Pterygopalatine.
Is that the only correct answer?
No, submandibular.
If you notice closely, axons from cranial nerve 7 innervate not one, but actually 2 named ganglia. One of them, as you guys mentioned, was the submandibular. And the other one sounded a whole lot like one of those alphabet soup terms we're going to need to know. And what was that alphabet soup term again?
Pterygopalatine.
Excellent, pterygopalatine. Now notice once again, these alphabet soup terms tell you everything about it. When I say Terry, what does that remind you of?
Is it the pterygoid or
Well, again, especially with little kids. If a little kid starts to say Terry, how are they finishing that?
Dactyl.
There you go, pterodactyl. Because a pterodactyl is a very special dinosaur. Yes, I know it's not a dinosaur, but let's pretend it's a dinosaur. What's so special about a pterodactyl? Okay, what's the second thing that's special about a pterodactyl? Because what's the first thing that's special about a pterodactyl?
is it its claws?
No.
Flies.
Yeah, okay, obviously. The first thing that everybody remembers is that they fly. However, what do they look like? What's distinct about the way they look? You guys need to watch Jurassic. Yeah, huge, massive beak. And that's what pterygo refers to. Pterygo refers to the beak. So notice our pterygopalatine, pterygo meaning the beak, means that this is going to innervate our mucous membrane of the nose and the nasal cavity. So let's say nasal cavity. Excellent. So that's part of the palate, roof of the mouth. And this also innervates our lacrimal gland. What does our lacrimal gland do?
Right. Tears.
Tears, absolutely. And again, these are just parasympathetic nerves that go from there. Whereas our submandibular innervates what?
Tongue. Salivary glands.
Salivary glands. Now it turns out there are actually 6 total, 3 pairs of salivary glands that you have on both sides. And it turns out this submandibular gland doesn't control all of them. There is another ganglion that also helps to control the salivary glands. And what is that second ganglion that helps to control salivary glands?
Otic.
Otic ganglion. Excellent. And I think I can fit that up there, which allows me to do this. And is the otic ganglion innervated by the facial nerve?
No.
No, what's it innervated by?
Central nerve 9.
Yeah, our glossopharyngeal. Excellent. So notice for this exam, I'm not going to make you distinguish which of our 3 salivary glands are innervated by the submandibular and by the otic. You just need to know that these 2 collectively, both of these, the otic and the submandibular, innervate our salivary gland. So notice if we go back to our picture, we see these 4 pathways. Cranial nerve 3 innervates the ciliary ganglia, where it synapses on our postganglionic neuron, which innervates the eye. Cranial nerve 7 innervates the pterygopalatine, which innervates the lacrimal gland and also the mucous membrane of the nose and the roof of the mouth, but also innervates the submandibular gland, which innervates salivary glands. Cranial nerve 9 innervates the otic ganglion, which innervates the other salivary gland. 4 pathways, 4 named ganglia. And notice for the rest of the pathways that are shown here, pretty much everything outside of the head. So in fact, we can take our highlighter here. Basically, everything else is going to use terminal ganglia. So everything from our vagus nerve use terminal ganglia. And we can't forget about the stuff coming from our lateral gray horn, which are going to use terminal ganglia as well. All right. Questions on these first 4 pathways? All right, excellent. Remember, we mentioned that in our parasympathetic nervous system, our vagus nerve is responsible for about 90% of the parasympathetic output. And that's not surprising when you see that so far all we've done is gone to the head. Our vagus nerve is going to go to many terminal ganglia, where they then form parasympathetic nerves. That innervate most of the organs of the ventral body cavity. So before we just use that in generic terms, but now we can be more specific. Our vagus nerve innervates the heart. That's not how you spell heart. Innervates the lungs, innervates the stomach, innervates the liver, innervates the gallbladder, innervates the spleen, innervates the pancreas, innervates all of the small intestine, and innervates the proximal part, large intestine. Again, remember, cranial nerve 10 is our wanderer. And that wanderer goes everywhere. Notice when we go back to our octopus, lo and behold, we see that axons from cranial nerve 10 go to the heart, go to the lungs, liver, gallbladder, stomach, pancreas, spleen, small intestine, and proximal part of the large intestine. And notice at every one of those locations, it either synapses right outside or actually in the organ. So none of these places of synapsing have a specific name. So they are just solely simply referred to as terminal ganglia. So the vast, vast majority of the pathways to the organs of our body just simply use terminal ganglia. Only the 4 in the head have names. All right. Questions on that?
Nope.
Excellent. All right. Now, remember, as we mentioned, most of the preganglionic neurons are found in nuclei associated with our cranial nerves. But of course, as we know, most means not all. So there are a few that are located in the sacral spinal cord. And of course, specifically in the lateral gray horn of the spinal cord. And of course, their axons are going to leave the spinal cord. And when they leave the spinal cord, this time they're not going to form cranial nerves. They're, of course, going to form spinal nerves. But as we know, spinal nerves branch almost immediately to form the dorsal ramus and the ventral ramus and the rami communicantes. And in the case of the parasympathetic nervous system, they become something we call the pelvic splanchnic nerves. Now, some of the textbooks, I don't remember if your textbook is one of them, have slowly started just referring to these as pelvic nerves. And that is an acceptable name. I put the name pelvic splanchnic here because you'll see why in just a moment when we get to the sympathetic pathways. But these pelvic splanchnic nerves then innervate our terminal ganglia, where of course then our parasympathetic nerves have to innervate something. And the question becomes, what's left? What is it that these sacral spinal cord structures innervate? And it turns out the specific effectors for these sacral portion of the spinal cord is the distal large intestine, primarily the rectum and the anus. right? Because as it turns out, defecation is a sacral spinal cord reflex, but also the urinary system, organs, or let's say urinary organs, the bladder, the ureters, and even a little bit the kidneys, and the reproductive organs. So notice if you want to pee, poop, or cum, you basically have to use the sacral portion of your spinal cord for defecation, urination, and reproduction. And again, notice if we look at our illustration, lo and behold, we see that. The nerves that start in the sacral spinal cord become the pelvic nerves. And again, don't forget that term pelvic splanchnic. It is totally okay to not use the term pelvic splanchnic. But when we get to the sympathetic, there are going to be splanchnic nerves. And that's how I tell them apart. Parasympathetic use P pelvic splanchnic. Sympathetic use S splanchnic nerves. So plain splanchnic nerves, splanchnic S, sympathetic. Pelvic splanchnic, P pelvic, P parasympathetic. So that is how although I guess pelvic and splanchnic, that still works for that as well. But both are acceptable terms. And again, notice their axons go to the rectum and anus, to the urinary system, and to the reproductive organs.
I'm sorry, could you repeat that?
Again,
Splanchnic.
we'll get to the sympathetic in a minute. Don't worry about the sympathetic. The reason, like I said, here the reason I mention this is your books and many textbooks are starting to go more and more to just referring to these nerves as pelvic nerves, the ones that are formed by the preganglionic axons coming out of the sacral spinal cord. They are also, though, often referred to as the pelvic splanchnic nerves. When we get to the sympathetic nervous system, we'll learn about nerves that are just called splanchnic nerves. So how do you tell the difference between pelvic splanchnic and splanchnic? Well, splanchnic starts with an S and it's in the sympathetic pathway. Pelvic splanchnic starts in the P and it's in the parasympathetic pathway. So that was the only point I was trying to make. Tell the difference between the splanchnic and the pelvic splanchnic is pelvic's got P, pelvic parasympathetic. Okay. So that was all I was trying to explain.
Got it. Thank you.
It's very simple, straight. Well, okay, maybe not simple, but compared to the sympathetic, you'll see it is very simple. A very linear relationship between where we are in the central nervous system to how we get to the effectors. Right? We have those 4 nuclei in our cranial nerves, 3, 7, 9, and 10. Only 4 named ganglia, all associated with the head. And technically, 10 doesn't innervate one of them because 7 gets 2 of those. Here we see another nice illustration that shows this. Again, cranial nerve 3, ciliary ganglia, smooth muscle of the eye. Cranial nerve 7 goes to the pterygopalatine, mucous membrane of the nasal cavity, the roof of the mouth, and also to our tears. But it also innervates the submandibular, which innervates some of our salivary glands, whereas cranial nerve 9 innervates the otic and innervates our other. Notice here they've given us the names, but again, you don't have to worry about that. You just have to know that these 2 ganglia innervate our 3 salivary glands. Here we see that nice vagus nerve with 90% of our parasympathetic output, and it is that big, huge wanderer. Heart, lungs, liver, gallbladder, stomach, pancreas, spleen, which I don't see here, but it is, proximal part of the large intestine, and the entire small intestine. Notice all of these have long preganglionic axons that go either into or right next to the organ that it is innervating. And again, even though this is 90% of the output, none of the ganglia have names. They're just called terminal ganglia. Technically, if it is within the walls of the organ, they will sometimes use the term intramural. If you think of intramural sports, these are sports done within the walls of the campus. So if it's in the walls, they sometimes use the term intramural. You're welcome to use that, but terminal ganglia is a simple generic term that is perfectly acceptable. So don't feel like you have to use intramural. But I mention it because, again, your book does, and I don't want there to be confusion. But we cannot forget about the sacral part of our craniosacral nervous system. Here in the sacral part, we have neurons, preganglionic neurons, whose cell bodies are located in the lateral gray horn of the sacral spinal cord. Their preganglionic axons come out and form those pelvic splanchnic nerves, or just pelvic nerves, and they go to the rectum and anus, they go to the urinary system, and they go to the reproductive system. And notice, as we said, you don't cry and salivate at the same time because we have different pathways. A different route to the liver than there is to the stomach, than there is to the bladder, than there is to the eyeballs, than there is to the salivary gland. Local discrete effects with a local discrete pathway. Questions on that? All right, excellent. Silence means that I totally understand that. And
Can you go back to the whiteboard for 1 second, please?
No. Yeah. There we go. So like I said, easily an essay question. I could give you any possible effector and ask you to describe the pathway for our parasympathetic to get there. All right, so if I said, for instance, describe the pathway that our parasympathetic nervous system would use to produce tears, what would that pathway be?
Ganglion,
Go ahead, Emily.
ganglion for cranial nerve 7, cranial nerve 7, the station.
Yeah. So it's cell. So again, remember, you got the right idea. Absolutely. The cell body is located in the ganglion of cranial nerve 7. Its axon forms cranial nerve 7, the facial nerve. Its synapse is where?
Pterygopalatine ganglion or the submandibular ganglion. Oh no,
Well,
sorry.
no, we said, remember, we said lacrimal glands.
Yeah.
So yeah.
Pterygopalatine.
Pterygopalatine. It's then its axon. Or let's say it this way, so postganglionic axon, whereas this was our preganglionic axon, forms a parasympathetic nerve, and then it innervates the lacrimal gland. Excellent. Just that simply. So yeah, this was our preganglionic cell body located in that brainstem in the ganglion for cranial nerve 7. Its axon forms cranial nerve 7. Its synapses on our postganglionic axon in the pterygopalatine ganglion. That postganglionic axon forms a parasympathetic nerve which innervates the lacrimal gland. And what neurotransmitter would it release?
Acetylcholine.
Acetylcholine.
Excellent. What neurotransmitter does this one release when it synapses?
Acetylcholine.
Excellent. What is the effect of the acetylcholine here?
Excitatory.
Was it excitatory or inhibitory? What one was it?
Excitatory.
Excitatory. Excellent. What is the effect of the acetylcholine here on the lacrimal gland?
Excitatory.
Yeah, to produce more tears. Now, the good news is for our parasympathetic, it always releases acetylcholine here. It always is excitatory for our preganglionic neuron. The good news is our postganglionic neuron in our parasympathetic is always going to release acetylcholine. So the only thing that could possibly be different in the pathway would be what the effect of that is. So on the lacrimal gland, it's excitatory. On the salivary gland, it's excitatory, right? But what about on the heart?
Inhibitory.
Inhibitory, right? So notice that. So that really, when it comes to the neurotransmitter and effects, the only thing that's going to be different is what the effect of that acetylcholine is on the ultimate effector. So easy pathways. And again, it may not seem easy, but trust me, compared to what we're going to be doing in the sympathetic, this is much, much easier. All right. If we are comfortable with this, or at least have some basic understanding of this, we can move forward. So any questions on this? Because like I said, it gets worse from here. All right, excellent. Then let's switch from our parasympathetic to our sympathetic. Again, remember, parasympathetic had these discrete innervations. And again, notice beautifully now that we have a better understanding and we're looking at this octopus is making a little bit more sense. Cranial nerve 3 is going to the eyeball. Vagus nerve is going to the heart and the lungs and the stomach and the pancreas and so on and so forth. Sacral going to the bladder and the genitals, right? So even though this is a very simple looking diagram, notice here they're synapsing outside because those two are named ganglia. These are inside the organs. They're just terminal ganglia. It's actually showing us what we have just learned. And unfortunately, over here, it's showing us what we're going to learn as well. Our sympathetic is a bit more of a mess. As we mentioned, it starts easy, so that's the good news. It starts easy in that all of the preganglionic neurons, their cell bodies are located in the T1 through the L2 or L3 region of the spinal cord, which is why anatomically we call this the thoracolumbar branch. So if we were to go back to our whiteboard, when it comes to our pathway, we still have to worry about the same types of stuff. The good news is it starts easy. All of our preganglionic neurons are located in the lateral gray horns of spinal cord sections T1 through L3. No, that's not a 1. But that's the last part of that is easy. We'll worry about what the axons of these form in a minute. And instead, we're going to look at, because again, remember, this is the location of the preganglionic neuron cell bodies. We also need to care about the locations of the postganglionic cell bodies. And we know these are going to be found in ganglia. And it turns out there are two types. There are two types of sympathetic ganglia. The first are what are known as chain ganglia, or they're sometimes referred to as trunk ganglia. So either of those are acceptable terms for these, chain ganglia or trunk ganglia. And the second type are what are called, oh, hold on, collateral ganglia, or what are also sometimes referred to as the prevertebral ganglia, whereas these chain ganglia are also sometimes referred to as the paravertebral. So yes, three potential names for this first type of ganglia, because of course anatomists hate you. There are lots of these, and they are paired ganglia. What do you think that means?
Right and left.
Yeah, there are two of them, one on each side. Here, there are only three collateral ganglia, and they're single ganglia. And that's actually where they get their name paravertebral and prevertebral. They get their name by the relationship to the spinal cord. Here is the spinal cord. The chain ganglia, which looks like I need to make pink for consistency, are located to the sides of the spinal cord. So since they're to the side of the spinal cord, paravertebral, like things that are parallel are to the side. Whereas the collateral ganglia are in front. A single one right on the midline in the front, and so they're in front of the vertebral column, so they're prevertebral. So two potential locations. We'll see this in the picture in just a minute, and then these collateral ones to the side. And not surprisingly, they're going to be different in the effectors they innervate. Our chain ganglia innervate basically the head. They innervate the skin and limbs and all the organs above the diaphragm. And so what does that leave for our collateral ganglia to innervate?
Under the diaphragm.
Yeah. Those in the abdominal pelvic cavity. Now, again, remember with our sympathetic nervous system, these are our big global effects. with many connections. So these are more general pathways because there's a lot of overlap and a lot of mush and gush in this system. But in general, these are the pathways. Let's take a look at the pretty pictures and go through this again. Notice here, this simple illustration shows us the chain ganglia to the side of the spinal cord, and notice they're innervating the head, the heart, the lungs, salivary glands, things in the head, and the skin. Whereas notice we also have our collateral ganglia here, whose axons go straight to there to synapse, and they go to the organs below the abdominal pelvic cavity. Liver, stomach, spleen, kidneys, adrenal gland is down here, you know, small and large intestine, and all those other things. Notice, again, we mentioned these are single and these are paired. They're off to the side here in our illustration, but remember, they're normally located here on the midline. We just, if they put them in front, that'd be confusing. So that's why they put them off to the side and call them the collateral ganglia when we do that. Here, we actually see a better illustration of this, how this works three-dimensionally. Notice, as we talked about before, our ventral root and our dorsal root come together to form our spinal nerve. Our spinal nerve forms the dorsal ramus and the ventral ramus. And remember, we also said it formed the rami communicantes, the white ramus, which is lateral, the gray ramus, which is medial. And remember, we talked about how these are primarily sympathetic pathways, because notice where they go, to this row of ganglia that are to the side of the spinal cord. Here we see the chain going down one side, and lo and behold, here we see a bit of the chain going down the other side as well. Notice other axons are coming here towards the middle, and even though they haven't drawn it on this illustration, here, sitting in front of the spinal cord, where would be where our collateral, ganglia would be located. So this is how we see why they're called para and prevertebral. Pre is in front, para are to the side. But again, you're welcome to use the terms chain and collateral. Those are totally fine and acceptable terms. You've seen this one. Let's jump to this one here. Notice what's nice about this illustration is here we see how the chain ganglia are indeed paired. We have one parallel line going this way, one parallel line going this way. However, I do want you to notice something interesting happens. Not surprisingly, the ganglia are named by the region of the spinal cord they're associated with. So the top ones are cervical, and then thoracic, and then lumbar, and then sacral, and then coccygeal. Notice we have two coccygeal ganglia, one from each side. But what's interesting about them is they actually fuse together. When they fuse together, they form a special structure called the ganglion impar. So while the chain is two parallel rows, it really looks like a big U, where you have the two parallel sides and then they come together in the center. Now, as I mentioned, you have one coccygeal. Almost everybody has three cervical, but the number of thoracic, lumbar, and sacral will vary from person to person. So if I randomly grabbed one like this one right here, would you be able to tell right off the top of your head whether that was T11 or whether that was L2? No. Right? So we're not going to worry about naming any of them except these four, because almost everybody has 3 cervical. They are given special names: superior cervical ganglion, middle cervical ganglion, and inferior. And notice I did say most, because in a couple people, in a few people, only about 7% of the population, these two fuse together. The middle and inferior fuse together. Functionally, no difference on how it affects your body. Just morphological, individual. And then our coccygeal, which again, the 2 of them fuse together to form that ganglion impar. So other than those 4, you don't need to be able to identify them individually. Although we do have some cool models in the classroom. Luckily, we can see some pictures of them here. Notice here is a model a big plaque found in the classroom that we affectionately refer to as the baby back ribs. And if you look closely at this, you can see the spinal cord and the nerves coming out. Notice great example of the brachial plexus, the lumbar and sacral plexus. It does a really nice job of showing us the conus medullaris, the caudae equina, a lot of the anatomy. But if we look closer, you'll notice, lo and behold, 1, 2, 3 big ganglia located right there, and then a bunch of additional ganglia all along the sides of the spinal cord. Here we see those chain ganglia, and sure enough, superior, middle, and inferior cervical, and then all the rest. Notice this does come all the way down, but you can't really see the ganglion impar. So this model doesn't show that, but it would come back up and go along the other side. So we see that nicely there. One place where we see the ganglion impar pretty nicely is the chart that's in the classroom. Notice here, the nervous system chart, all the nerves are in yellow. But if you look closely, there are some white spots. 1, right, 2, and the third one is actually right here, although you can't see it that well in this picture. 3 cervical ones here, but notice the chain continues on down. Here, we actually see the collateral ganglia. 1, 2, and 3. The 3 collateral ganglia that are here in the front. And I think, if I remember correctly, they have, yeah. Again, I wouldn't show you this at this magnification, but we're sitting here playing with it right now. So that's all we're doing right here. But if you notice, our chain actually comes down, and then right here, where it starts to go up the other side, we can actually see where the 2 coccygeal ganglia come together to form that ganglion impar. So aside from the pictures in the textbook, there are some models, there are some charts that will show some of this stuff you are responsible for. All right. And like we said, these chain ganglia innervate the head, the limbs and skin, and all the internal organs above the diaphragm. All right. Questions on this? All right, I know it's a bit overwhelming, but we still have a lot of work to do on the sympathetic. We need to talk about all the structures that are formed by the axons. All the structures that are formed by the axons of the sympathetic preganglionic neurons. And because these are so much closer to the central nervous system, we are going to need to identify the structures that are formed by the post. axons. As I mentioned, asking you about a parasympathetic pathway is definitely a potential essay question on this exam. But what we will see tomorrow is that there are 4 main pathways in our sympathetic nervous system. To get to any of the organs of the body, there's primarily 4 main paths you can take. And I guarantee you with 100% certainty that one of those pathways will be an essay question on your exam. The good news is we will go over those pathways together tomorrow. The better news is your textbook does a really nice job of describing at least 3 of the 4 pathways. It does mention the 4th, but it doesn't emphasize it as much as it should. So if you haven't done so already, I strongly encourage you to look at the lecture slides, to look at the textbook for this part. And tomorrow, we will finish the rest of the anatomy we need to know, and then we will describe the 4 pathways. And I guarantee with 100% certainty that you will get an essay question that says, identify the pathway of the sympathetic nervous system for it to innervate X, heart, lacrimal gland, stomach, right? Bladder, whatever it is. And you're going to have to describe all the structures that are the neurons are located in, all the structures formed by the axons, what neurotransmitters they release, the effect of that neurotransmitter, all of it. Guarantee it. So come prepared, and we will work on that tomorrow. All right. Questions on that? All right. We have done the anatomy for our parasympathetic nervous system. We've done our parasympathetic pathways. We've done our comparisons between our autonomic and our somatic motor, and our comparisons of the sympathetic and parasympathetic. So we have covered a lot of ground today, a lot of material, but we still have a fair amount to do tomorrow. Not quite as much, but especially these pathways can get pretty complicated. So we want to make sure we have plenty of time to spend on them. And then we'll sum this up. The end part of this, really, like we saw before, isn't going to be a lot of new information. We'll be synthesizing information that we've already talked about, maybe adding a few fancy labels to things, but it won't be too much new stuff. The majority of the new stuff is going to be these 4 pathways, but you can come prepared. All right. Questions on any of that? All right. We worked long and hard. It's a little bit early, but I don't want to get too far into this anatomy. I want it to be stretched fresh as we go through the anatomy, so we can go straight from finishing the anatomy to the pathways and have it make sense. So I think this is a good natural stopping point. So that is all that I will cover for today. All right. Any questions?
No.
All brain dead. Excellent. That's exactly how I like you. I get to do that one more time to you, and then we are done. So excellent. All right. So study hard, enjoy your day, and I will see you tomorrow morning. Take care,
Thank
guys.
you.